Thank you, Dr. Kastner, for the warm introduction. I also want to thank Chip and the foundation putting together every few years. These meetings have always been amazing. So it certainly is an honor for me to present this talk, but this is not about me, except for 2 slides I'll talk about me, but otherwise this is presenting on behalf of the entire Data2 Foundation Consensus Committee that really made this work possible. Okay. These are my disclosures. I am on the SAB. I do some writing for UpToDate. I have some fundings that are not relevant for this talk, and I cannot do splits. So for really the trainees and audience, I like to reflect on how I got here and how I started working on Data2. And this was a week in my rheumatology fellowship, Boston Children's, and I was consulting a 2-year-old girl with recurrent fever episode. I remember this was a Sunday, and I give a lot of pushback, right? We don't do PFAPA consults on Sundays. But she had more than that. She had anemia, she was not gaining weight, she was just not quite well. And a number of months later, she had this episode where you can see the picture on the screen here, a CT scan that showed pretty impressive hemorrhage in the brain. She was rescued quickly by ED physicians as well as the neurosurgeons. And pretty quickly, Gigi Norangelo, who most of you know, was at Boston at the time, among others, suggested, could this be Data2? And I connected with Mike at Duke, and Ivona at NIH, send them some packages with the patient samples. And 2 days later, Mike called me on my cell phone and said the patient's deficient in ADA2. A couple days after that, Ivona called me and said, well, we found one of the mutations. And a couple days after that, I got a call from Dan Kastner teaching us how to manage this with TNF inhibitors. So all of this happened in a week. And I thought, wow, this is amazing. And all I had to do was send 2 packages by FedEx, and everything just happened and fell in place. But this is not an accident, right? This is not one case. This is what these people do every day. And these are people sitting next to you. This is what they do every day. And this is what makes this a very amazing community that I've grown to love. And with the permission of the family, this is now our patient. She's 10, doing very well on Enbrel, have no symptoms. And of course, this is Mom, who is also sitting in the audience as VP of the Data2 Foundation. So really an amazing story, and this is how I got here. All right, so let's talk about consensus. This is an effort from 37 clinicians from 18 countries, representing 12 adult and pediatric subspecialties, as well as 3 patient representatives. It was published in May of this year. Today, I won't tell you exactly about the individual statements. We'll talk about how we got there as a group. As you all know, there are many challenges for Data2. Right? I'll list some of those here. First, in general, for rare diseases, there's a lack of knowledge and awareness, right, across the field. Specifically for Data2, the phenotype can be quite variable, and even for patients within the same family, they have very different clinical manifestations. We have limited availability of diagnostic modalities. Even though we have an enzymatic testing that's available, only few places can do this across the world. And most importantly, we have very limited understanding of the pathophysiology. Right? This is the reason why we're all sitting here today. And all of this adds up to inconsistency in terms of diagnostic evaluation and the treatment approaches. And if we don't agree among ourselves, how are we going to teach a wider audience about this condition? So 3 years ago, in the 2020 virtual meeting, the 3rd international meeting for Data2, the theme was driving towards consensus. And the goal was to develop a consensus approach for diagnostic evaluation and management of the disease. Chip and the foundation appointed myself, Amanda Umbrella from NIH, and also an intern, Brad Davison at Vanderbilt, who's a PhD student working with the Data2 Foundation to lead this effort. So that's great. We can get started. And we had weekly meetings for a little while to figure out, oh, how are we going to do this? Well, that's easy. We can just look at what other people do for other rare diseases, and we'll just follow their suit. Apparently, there aren't that much available out there. So we really have to start from the beginning. And these are some of the key questions we had to figure out. Who should we involve? What questions should be addressed? And what questions can be addressed based on available evidence, which, as we know, is not that much. Who's the intended audience for this? And what's the method we should be used? The term methodology was new to me. I have no idea there's a whole methodology behind this thing, and there's actually a profession called methodologists to do these things. So it was a big bit of a learning curve in the beginning. So we needed help, right? We wanted help from different people that are involved in this disease. So we wanted help from neurologists, we wanted hem-onc doctors, we wanted immunologists, rheumatologists, and geneticists as sort of the major core group. And with that, the Data2 Foundation provided a list of names, 4 to 5 experts from each of these work streams that we called. And everybody was very enthusiastic in terms of participating in this process. Then we have to figure out, well, what are we going to address in this consensus? I mean, there are a lot of nice questions. Then the idea came to us, and well, why don't we take the questions from the people that really care about this? and that's all of you, right? People that sat through the first 3 Data 2 conferences. There's a whole list of Q&A from the clinicians, from researchers, as well as the families. So we took those questions as the one that are most pertinent to our field and categorized them into topics of diagnostics, screening, treatment, and long-term care. And I'll show you some example here. So this is a list of questions we've reformatted. Many of them overlap, so we put it in the right context. And so diagnostics testing for people that are suspected of Data 2, is the enzymatic test better or is genetic test better, right? Which one do we do first? How can you exclude a diagnosis based on the genetic testing? And can you exclude diagnosis based on enzymatic testing? And how do we define carriers? So these are all really important questions that are proposed by the crowd in previous meetings. After that, we did a very exhaustive literature review. We looked for all the abstracts that are relevant to ADA2 and Data 2. And finally, we selected 42 full articles published by the audience here, right, that'll be relevant to the research question we're trying to answer. Brad Davidson, our intern, nicely put together an Excel database with all of these articles stratified by their primary field of interest and distributed to everyone on the committee. And then we also needed some expert input to people that actually do these a lot more than we do. Particularly, I want to acknowledge Dr. Reem Mustafa for providing early guidance with this process. Then going through some of these articles, we have to grade the evidence, even though we know that we're not going to have any randomized trial in this condition yet. So we used the Oxford system, which is widely used for consensus methodology, which provides a level of evidence based on the literature available, as well as a grade recommendation based on the consistency across the different studies. So this is the framework we put together in the beginning. And so we wanted to synthesize questions from these meetings, from literature review it, as I mentioned. We drafted preliminary statements based on these 4 categories. And then we had our first virtual meeting. It was a bit of a challenge because these experts are spread across many time zones, right? So we made it work. And we distributed these statements to the different expert work streams. Okay, so particularly questions that are relevant to neurology, they go to the neurology work stream and so forth. And we asked everybody to give us feedback, and they gave us a lot of feedback, which is very nice. The revisions, we had some additional statements. We took away some that they thought was not good enough, and then they also evaluated our evidence grading in the process. So we got feedback from everyone, and then try to put together and make sense of the next version of this. And quickly we realized what we're missing is a strict definition of disease phenotype. And that made it difficult because it's really hard to provide specific statements for a condition where you have so many different disease phenotype. Right? So we added disease phenotype in its next edition, and that was around the time where COVID vaccine became available. So we had suggestions we should add recommendations about vaccination. And then we had so many recommendations or statements regarding management, and we had to divide it into initial management versus long-term management. And then it goes back to the work stream again for another round of revisions. And everybody reviewed it, gave a lot more changes, and then finally we prepared 32 of these statements for voting. Before we voted, we also wanted to increase global representation, so the foundation invited 10 additional expert clinicians from around the world, and also 3 patient family representatives. This is important because we really want to make sure that the questions we're addressing is what the families and the patients want to know. Okay. Then came to voting. This is the 9-point Likert scale, widely used for this type of methodology. Everybody get to put a number for each of these statements, whether you strongly agree or strongly disagree. This was done through a web survey set up by Mary Mackley and the Data 2 Foundation. And everyone was given the option to abstain. So if a question is not pertinent to your expertise, you're welcome to not answer those questions. We also provided a free text box with each of these items to let people explain why they voted a certain way, particularly when they disagree. Is it because of the concept of the statement, or is it because of certain wording, so we can make improvement? Then we calculated the disagreement index. I'll show in the next slide. And 30 to 32 statement passed the a priori disagreement index threshold that we put in the first round. And we sent the revised statement, the last 2, back to the committee and also asked them what other topics should we address in the future, things that we don't understand well. And after the second round, we reached agreement on these statements, and then we start discussion drafting the manuscript. You don't have to belabor the math here, but I really have to learn the methodology in terms of math behind this. And how do you calculate when people agree or disagree? And apparently, it's more than just a mean and standard deviation, because that tells you the spread, but doesn't give you the dispersion. So for example, when you have most people agree and a few people that strongly disagree, this methodology allows you to see that. So there's a sort of a fancy way of figuring out this disagreement score, and we were able to reach agreement on these after 2 rounds. Then the committee also had a lot of input in terms of what should we do in terms of publication, right? So the idea was to target a general audience of really general practitioners across the world, make a language that's not field specific. It should go to a general medical journal, so not a subspecialty journal to any of the subspecialty involved in this process. It needs to be open access, so anybody with web access across the world can pull it up. And with those in mind, we agree on a pre-submission inquiry to JAMA Network Open, and the editor gladly accepted our inquiry. The paper was completed, went to review, and was accepted after a couple of months. And this is the end product that was published in May, at the end of May this year. At JAMA Network Open, on the right here is just all metrics that measures how people, how well your article is being cited and how many people tweet about it and all these things. And apparently it's in the top 5% of our research articles, which is great. And we're also very grateful for Ron Lexer on his very insightful editorial that accompanied our article that really spells out the importance of doing this, not just for DATA2, but for rare disease as a whole. Again, I won't talk about individual statements because it's easily available through a net search, but this is just to show you what these statements look like at the end. There are tables looking at this one is the diagnostic evaluation for DATA2. We talk very specifically about the role of enzymatic testing and genetic testing. Each of those come along with level of evidence, level of agreement, and the disagreement index among the committee during the voting process. Then the next slide is on management. That's another table. And this one, we really prioritize the treatment approach that we should be using, right? So TNF inhibitor, we made it very clear, is a treatment of choice, right? So as a first item on this, so everyone can see it. Even though the level of evidence isn't great, right? This is primarily based on large case series. So we also have to acknowledge our limitations in this process, right? So we know coming in that this is a rare disease. We don't have a ton of rare evidence beyond many of the case series published by this crowd here. So it's based on very little evidence, and we avoided using the term recommendations and guidelines because we're just not quite there yet. And this is what ACR-ULR is doing, the points to consider also for type 1 nephropathies and inflammasome diseases, where these are things that we should consider and pay attention to, but not quite yet mainstream consensus. We understand that some statements have limited sensitivity and specificity, specifically when it comes to thinking about screening. Who do we screen for DATA2? And at this point, we want to err towards over-screening than under-screening, right? Because we clearly know that there's a lot of underdiagnosis of this condition. And we couldn't address everything. There are a lot of important and very popular questions that are not directly addressed. But it's important that we start somewhere, right? So this lays to the groundwork for future efforts to do more consensus that will be more precise for the field. And really have to get input from all of you today to get to there. So I want to outline some of the unanswered questions, and this is a really good segue in terms of what we'll talk about today. Right? These are questions that we were unable to address or just didn't have any evidence to go after. So is it possible to predict phenotype based on genotype? Does the phenotype change over time? Right? And I know some of the recent data to show that there are a few patients that could change their phenotype. What do you do about symptomatic carriers? Right? There are a couple of presentations today that will address this topic. How about the pre-symptomatic or asymptomatic individuals that we know have DATA2? Do they all require treatment? And how about patients with purely hematologic immunodeficient phenotype? Do we give them all TNF inhibitors? Right? Even they have a profound risk for infection already. And patients with the need for transplant but have no donors, what should we do? Do chimerism matter in terms of the outcome? And how about biosimilars, right? Many parts of the world rely on biosimilars for TNF inhibitors. Do they work as well as originator TNF antagonists? How about enzymatic therapy, which Chip just mentioned? And how about gene therapy? I know we have a couple talks later today to talk about this. So really a lot of unanswered questions that we hope to address during this conference. And now, Andrew, this is my other slide about me, right? This is my reflection from working through this process. I felt it was a really amazing and humbling process. Amanda and I, we learned so much together in this process. And coming in, I have not done consensus before. I thought we're just going to do a couple of Zoom meetings, we vote a couple of times there, we're done, right? But no, right? Methodology is a separate animal, right? I do a lot of clinical time, I do my basic research lab in the laboratory, but this is a completely new experience. Because there's not a lot of precedence behind this. We have to adapt to changes at every step. Like a lot of these things were not planned. We were hoping that there are a lot of other articles on rare disease that will be able to guide us, but it turns out that at the time we didn't have any. It was great teamwork. Amanda, really appreciate your work. Brad really helped a lot in a tremendous process in getting us started. And I also really appreciate the mentorship around the crowd. And I'm calling out a few of you guys here. So Paul Brogan, Anastasia Ozen, Marco Graterno, Isabel Mate. all of you guys have done so much more of this than we have, and I really appreciated their honest opinion, and not just saying, oh, you're doing good, but really an honest opinion about this is not how you do it. Right? It really helped us a lot to put this in context. And also proud to say that the statement is very helpful, right? So it's silly in this country when we try to apply for insurance for TNF coverage for patients data 2. I tell the patients, you can expect a letter next week, it'll be denied. Okay? You can expect another letter in 2 weeks, it'll be denied again after our appeal. Right? It's usually not until we get on the phone with somebody on a peer-to-peer to say, well, this is a bad disease, they may get strokes, and then they're reluctant to say, I don't know what you're talking about, but okay, sure. Right? That's usually how it works. But since we had the consensus, the last few times to do this, I get the denial, I send them back, this is the article, statement 5.1, okay? It's gotten approved every single time. I haven't had to talk to anybody on the phone, which is amazing. I know Paul is going through a very similar effort in the UK to try to get this approved widely, so I do think this effort will be very helpful to the community at the end of the day. And more importantly, this sets a foundation for future consensus effort. Right? This will not be the only addition. As we learn more about this disease from all of you, as we have sort of more precise evidence, right, to guide our management decisions. And I also hope that we can include more of you for this exercise. I know we're only a limited number of people involved, and I really hope that through the foundation we can get more and more people involved. And to end this, I just want to thank Chip and the foundation for having the confidence in us, even though we have very little experience going in. I think we got the job done, and I hope to work with all of you in the future to get the next edition going. Thank you for your time.